Hello? ഞാൻ കഴിഞ്ഞിട്ട് വിളിക്കാം. സോറി. കാണുന്നില്ലല്ലോ. സംസാരിക്കാനോ? അയ്യോ വന്നു. So, hello students, welcome to Nursing Guru, myself Soni. Today we are going to discuss the second semester subject. Two subjects we are discussing, that's applied nutrition and applied biochemistry. So first section is the applied biochemistry. So your exams are nearby, so we will help you to crack your exam by giving the expected questions of each unit of each subject. Not only the question, we will give you the answer key also. So you can refer the questions easily and you can study well. Shall we start? Okay. So in applied biochemistry, we have total 8 units are there. Each unit we will discuss the questions such as essay questions, short answer and very short answer questions. We'll move on to our first unit, that is the carbohydrates. So the expected essay question from this unit, the first one, classify carbohydrates and explain the digestion, absorption and metabolism of carbohydrates. So how you will go to answer this question? First you have to classify the carbohydrates. Mainly there is two type of classification is there. First one, chemical classification, and second one, physiological classification. Both these two classification again have subdivision, that is monosaccharide, oligosaccharide and polysaccharide. So you have to describe all the classification in detail. So classification is clear. And what is the next part? Digestion, absorption and metabolism. So you have to write how the digestion is happening, how the absorption, transportation and storage is happening. So mainly, you know, carbohydrates are broken down to provide glucose for the energy. So the main energy source is the glucose. So you have to describe regarding the glucose and the absorption. Clear? And the storage also you have to include in this essay part. And yeah, that's all regarding the first essay question. Moving on to the second essay, that is explain the factors regulating blood glucose level in the human body. So mainly glucose is a major source of energy. You may know, right? So mainly you have to include the factors. Mainly there are two factors that is regulating the blood glucose level. So the factors that two are, first one is the insulin, second one is the glucogen. So both these two factors you have to write in an elaboration. How insulin is acting on the blood regulation, and how glucogen is affecting on blood regulation. So this you have to include for the this you have to include in the factors regulating the blood glucose level. Okay, in this essay there is one important topic, that is negative feedback system. So negative feedback system also you have to include in your essay topic. Clear? Next we'll move on to the short answer question. Mainly we have 3 short answers expected, that is diabetic mellitus, GTT and role of 2 hormone. And what is diabetic mellitus? For this question you need to give a definition for diabetic mellitus and the classification. There is type 1 diabetic mellitus, type 2 diabetic mellitus. After that the symptoms, the management, treatment, also the complication, everything you have to mention for diabetic mellitus. Second question, GTT. GTT means glucose tolerance test. For this question how you will answer? define what is GTT and the procedure, indication for the GTT, how long it takes and the result. Also, you have to write principles of GTT. Clear? There are contraindications also. So, you can include the contraindication also for GTT question. Clear? Okay. Then move on to the third short answer question, that is name and indicate the role of two hormones that regulate blood glucose level. Okay. So, we have to mention two hormones. What are the two hormones? I will give you. First one is insulin and second one is glucagon. So, mention the names and elaborate. These two are how affecting for the blood glucose level regulation. Clear? So, moving on to the short answer question. We have three expected very short answer question. First one glycosuria, second HbA1c and third one hypoglycemia. Okay, in that you need to define what is glycosuria. Just give one definition like blood glucose level are so high, the renal tubules in the kidney cannot reabsorb it all. So like that, just define the glycosuria and you will get the at least 2 score from this question. And next one is HbA1c. What is HbA1c? That you have to mention, and the uses and the treatment, everything you have to mention for HbA1c. Third question, what? Hypoglycemia. What is hypoglycemia? We all know hypoglycemia means it's a condition. The sugar level is reducing. So you may all know the sugar level also. So these all are the expected question from our first unit, carbohydrate. Clear? Essay is classify carbohydrate in the digestion, absorption, metabolism. You have to include. Second question, factors regulating blood glucose level. Clear? And 3 short answer, diabetic mellitus, GTT, role of 2 hormones. And very short answer, it includes glycosuria, HbA1c and hypoglycemia. Clear? Okay. Then we'll move on to second unit. What is the second unit? That is lipids. From the unit lipids, we can expect the essay, that is digestion and absorption of the lipid. How you will answer for this question? Same as carbohydrate, you have to define what is a lipid and you have to answer how the digestion is happening and the absorption of the lipid. So you may all know the digestion, it starts from the mouth to stomach and it's going to as a bloodstream. Clear? So like that you have to answer for the question of lipids. Clear? That's the only one essay question from the lipid. Next is the short answer question. So we can expect 5 questions from lipid. First one is functions of essential fatty acids. If you get a question like functions, you have to explain at least 10 points functions, 10 points of function. Either you can answer like single word of 10 points, after that elaborate with the 2 sentences. Or you can also answer like 2 sentences of 10 points. Okay, either both 2 way also you can use to answer the functions. So at least 10 points you have to describe for a functions of lipid. I will tell you one function, that is this occurs in high concentration in the reproductive organs. So like that 10 points you can elaborate. Clear? Then we'll move on to the second question, that is metabolism of cholesterol. Okay, how the metabolism of cholesterol happen? First you have to write down what is metabolism of cholesterol, and in that what all are includes. Mainly it includes lipids and lipoproteins, and the functions of lipids and lipoproteins you have to write. Also there is 2 type of pathway metabolism is there. Like first one is exogenous pathway for lipid metabolism, second one is endogenous pathway for lipid metabolism. So this is a metabolism. Clear? For the metabolism of cholesterol there is 2, that is exogenous pathway and endogenous pathway. So both these 2 pathway you have to elaborate for the short answer question. Clear? And we'll move on to the Third question, functions of lipoprotein. So same like that. Mention what is lipoprotein and write down the function, at least 10 functions. So mainly lipoprotein that have classification. So you can include the classification to write the functions. Mainly we have 5 classification, that is chylomicrons, very low density lipoprotein, intermittent density lipoproteins, low density lipoproteins and high density lipoprotein. So mention the 5 classifications of the lipoproteins and elaborate the 5. Then you will get the score. Clear? So that the 5 times you have to be by heart. And we'll move on to the last question. Sorry, fourth question. That is ketone bodies. So what is a ketone body? Same like you have to define what is a ketone body and the uses in the body area. So first use it in the heart and brain. So elaborate use in the heart and brain and the production. Next, use in ketosis and the ketoacidosis. And you have to include the pH impact also in the ketone body question paper. Clear regarding the ketone bodies? Okay, last one is a MUFA and PUFA. What is MUFA and PUFA? MUFA and PUFA. There is a full form. That is monounsaturated fatty acid. That is MUFA. And PUFA means polyunsaturated fatty acid. So you can expect this question same in very short answer question. What is that difference between MUFA and PUFA? So mainly we can see in monounsaturated fatty acid, this is a healthy fat molecules. Okay, mainly we can see in nuts and also natural foods and seeds, olive, avocado. So mainly this source the monounsaturated fatty acid is presenting. But going to the PUFA, what is we can see? Mainly we can see 2 type of omega-3 fatty acid and omega-6 fatty acid. So that is a different. Also there is one more different is there. The unsaturated fatty acid that contain 1 double bonded carbon and atom in the fatty acid chain for MUFA. But PUFA there is 2 or more double bonded carbon atom. That is the main difference for the MUFA and PUFA. Clear? Okay. Next we'll move on to short answer question. In short answer question we have 5 expected question, that is Rother's test, difference between MUFA and PUFA, trans fatty acid, type of cholesterol and lipid profile. Okay. So this you can write down with 3-4 sentences. Okay. Rother's test means what? Rother's test is the test for ketone body. So that you have to be elaborate the procedure, the duration and the result. Okay. And difference between MUFA and PUFA we already discussed, that you can write for your examination with a column based answer. And third, trans fatty acid. What is the trans fatty acid? It's a fat that form of unsaturated fat and come in both nature and artificial form. So it have 2 form, nature and artificial form. Okay, like that you have to write down that question. And the fourth question, that is type of cholesterol. What are the type of cholesterol? Mainly we have 2 type of cholesterol are there. That is low density lipoprotein and high density lipoprotein. What is good cholesterol? That is high density. And the low density is the bad cholesterol. So you have to compare the 2 cholesterol. Last one is the lipid profile. What is a lipid profile? In a lipid profile, it includes the levels of total cholesterol, high density lipoprotein and triglyceride and calculate a low density lipoprotein. Okay, like that you have to answer for the very short answer question. So the second unit is clear. We can expect 1 essay question, that is a digestion and absorption of the lipids, and 5 short answer question, functions of essential fatty acid, metabolism of cholesterol, function of lipoprotein, ketone bodies, and MUFA and PUFA. This is MUFA and PUFA is important because you will get the very short answer question also. In very short answer, Rother's test, difference between MUFA and PUFA, trans fatty acid, type of cholesterol, lipid profile. Clear? You can also note down the questions and the answer point, so you can easily refer all the topics. We'll move on to the third unit, that is protein. From this unit we can expect one essay question, that is a classification and biochemical functions of plasma protein. So first we have to classify plasma protein, then we have to write the biochemical functions. Same as first give a definition for the biochemical, first you have to define what is a plasma protein and then only go for the classification. After definition, there are classifications such as albumin, alpha-1 globulin, alpha-2 globulin, beta globulins, fibrinogen, like that. So write down the names and elaborate each type in a paragraph. So you will get the score. After the classification, write the biochemical function. For the biochemical function, mainly you have to include the functions of plasma protein. What are the functions coming in the plasma protein? First function, protein nutrition, and second, osmosis, pressure and water balance. Third, buffering action. Fourth, transport of lipid. Fifth, transportation of the other substance. And sixth, blood coagulation. So total 6 functions you can include for a plasma protein. Clear? So like that you can complete this essay question. We'll move on to the short answer. We have 4 short answers we can expect from the protein chapter. First one, digestion and absorption of the protein. So write down what is protein and start to write with the digestion. How the digestion is happening. Mainly protein digests in the stomach and the protein digests in the small intestine. And where is the absorption is happening? In the jejunum and ileum. So like that explain the digestion and the metabolism and synthesis of the protein also you have to include. In that you can include a single molecule amino acid or free amino acid that are absorbed through the wall of small intestine. Clear? Like this you have to answer for the first question, digestion and absorption of protein. Clear? Next, we'll move on to the second short answer question, that is amino acid. What is an amino acid? Define what is amino acid and classify amino acids. Mainly for the amino acid, there are 2 classifications. The first classification have 2 subdivisions. Okay, we'll go by detail. First classification, that is based on natural requirement, and also classification based on mechanical rate also there. So both these 2 again subdivisions are there, essential amino acids and non-essential amino acid. For classification based on the metabolic rate, the sub classification are glucogenic amino acid, ketogenic amino acid. Yeah, these 2 are the sub classification. Other than this classification, one more is there, that is primary amino acid and non-primary amino acid. So this is the classification of the amino acid. Clear? You have to elaborate the classification in detail. Next, the third question, protein metabolism. So protein metabolism, you have to include the process of protein metabolism. What are the processes you can include in the protein metabolism? There are transcription, translation, post-translation modification. Yeah, these are the 3 processes of protein metabolism. So write down these 3 and elaborate the 3 processes. Clear? And move on to the fourth question, that is electrophoresis. So you have to define what is electrophoresis and the uses and the principles, everything you have to include. Mainly in the uses, there is 2 type of uses are coming. First one, used for forensic. It means DNA fragmentation and RNA fragmentation, separation of the PCR products analysis. Okay, and second one is a classification for application involves. In that it's involved lipoprotein analysis and urine analysis, like that. Clear? After that, mention the principles of the electrophoresis. That is the question answers, and you can also involve some more points, that is factors affecting the electrophoresis. Mainly there are two factors: inherited factors and external environment. So this also you can include for this question. Clear? Okay. So our short answer questions finish. Next is a very short answer question. First one, essential amino acid. So essential amino acid means we have to mention what all are the essential amino acids and the uses and how it support the human metabolism. Clear? Okay. Second is the inborn errors of amino acid. So write down what is inborn errors of amino acid and what all are included in that. In that phenyl alanine is there, triphosphate is there, and trizonin, everything is there. That also you have to include for your second question, inborn errors of amino acid. Clear? Okay, move on to the third question, that is aromatic amino acid. In that aromatic amino acid means it's a acid and this amino acid include the aromatic ring. Okay, you can answer by two sentences. And last is the fourth one is the proteinuria causes. Okay, what are the causes of proteinuria? Mainly there are causes that are harmless condition including dehydration or intense exercises, or more serious including kidney disease. So these are the main causes that can cause proteinuria. Clear? And the fifth question, that is hypergamma-globulins. So you have to define what is hypergamma-globulins. Mainly it refers to overproduction of more than one class of immunoglobulins by plasma cell. Clear? That is the definition for hypergamma-globulin. Okay, so that's the question from protein chapter. We'll move on to the next chapter, that is fourth one, clinical enzymology. In that we can expect only one essay, that is isoenzyme and clinical significance. In that you have to write down what is isoenzyme. Okay, it's also called as isomes. And there is a classification, that is LDH and creatine phosphokinase, and third one alkaline phosphate. So you have to write down these three and write down elaboration of the three. In that there is a common point, that is H and M. H mean the muscle, M mean the muscle, H mean the heart. You have to include the LDH1, LDH2, LDH3 and CPK1, CPK2, CPK3. Like that you have to elaborate the isoenzymes. So the essay is clear, no? We'll move on to the short answer, that is enzyme in the liver function test. So you have to write what is a liver function test and the enzyme. So mainly the enzymes are alanine transaminase, aspartate transaminase, alkaline phosphate, albumin and total protein, bilirubin, gamma globulins, lactate, dehydrogenase, prothrombin type. Okay, so these are the enzymes that is involving in a liver function test that you have to write for your examination. Clear? And the very short answer, that is the normal values. So normal values, it's already in your textbook. You can refer the points in We are also mentioned in the Nursing Guru app. So refer from that. It's in a column base. For it include the acid phosphate, alkaline phosphate, alane transaminase, and creatine kinase, lipase, lactate, everything is coming. So you have to by heart the normal values also. So sometimes they will ask one or two normal values. Clear? Okay. So that is only expected questions from clinical enzymology. We'll move on to our next chapter, that is the 5th unit, acid base maintenance. In that, the expected essay question is, what is the normal pH of blood? Describe the role of lung and kidney in the maintenance of normal blood pH. Okay, so first you have to write the pH. What is the pH? And the normal value of pH. What is the normal value? It is 7.4 or 7.35 to 7.45. Okay, once the pH is increased, that is known as acidosis. Once the pH is reduced, that is alkalosis. Okay, so you have to mention the metabolic acidosis, metabolic alkalosis, respiratory acidosis, respiratory alkalosis, also unconjunctival metabolic acidosis are there, unconjunctival metabolic acidosis are there. So you have to be mentioned all in a detail. So that is coming in the role of lungs and role in lungs and kidney. Okay, so once the pH effect in our respiratory system and renal system, the pH value change, it can cause several disease condition. Okay, I also mentioned it decrease means that is acidosis. Sometimes respiratory condition also can cause respiratory acidosis, COPD and emphysema. All these causes can lead to any kind of acidosis or alkalosis. So you have to be define the classification. Clear? And move on to the short answer. That is metabolic acidosis, arterial blood gas and acid-base disorders. Okay, the first one is metabolic acidosis. So define what is metabolic acidosis. What is metabolic acidosis? It is a too much acid in the body fluid. It means increasing the pH is the acidosis. Okay, and you have to mention what are the causes of metabolic acidosis, other causes and symptoms, diagnostic measures and the treatment. Like that you can conclude the metabolic acidosis question. Second one, arterial blood gas. Arterial blood gas means ABG, no? It's a test we are conducting to evaluate the pH and the blood gas in the body. Okay, so that also you have to be elaborate. And in that, one more thing is there, write down the indication for the blood gas analysis and the procedures. Also, the arterial blood gas analysis measures also you have to include it for the arterial blood gas question. Clear? What's the third question? Acid-base disorders. Acid-base disorder, there are certain disorders is occurring due to acid-base imbalance. So you can classify it as metabolic acidosis. So metabolic acidosis is happened by acid indigestion and decreased renal acid excretion, increased acid production. So these all can cause metabolic acidosis. And metabolic alkalosis. What is metabolic alkalosis? Mainly acid loss can cause metabolic alkalosis. Next, respiratory acidosis. Respiratory acidosis means mainly the hypercapnia that can cause respiratory acidosis. Next, respiratory alkalosis. Respiratory alkalosis means hypocapnia. And next is the compensatory mechanism, and there is a simple acid-base disorder also can cause in the acid-base disorder. Okay, and you can mention the symptoms also in this question. There are diagnostic features also for to detect the acid-base disorder that you can include it for this question. Clear? Yeah. Next we'll move on to very short answer. Very short answer question. Mainly we have 3 questions. pH definition, blood pH and blood buffer. Okay, we already discussed the pH normal value, that is 7.35 to 7.45. So that you can write down there. And second is the blood pH. Blood pH also you have to write down the blood pH values and the normal and the abnormal ranges. Then blood buffer. Blood buffer is a small topic. Within 2-3 sentences you can answer for the blood buffer. So clear? So that's only in the acid base maintenance. SA is pH of blood. Short answer, metabolic acidosis, arterial blood gas, acid base disorder. And very short answer, pH definition, blood pH and blood buffer. Clear? Okay, next we'll move on to our 6th unit, that is hemicatabolism. In that we have only short answers and very short answers. Not expecting any essay from this chapter. Most probably you will get the short answer, that is hemidehydration pathway. You have to draw the pathway of the degradation. So first you have to define what is a hemidegradation pathway. It means heme is a cyclic tetrahydrous that contain iron and commonly found as a prosthetic group of hemoglobin, myoglobin and cytochromes. Okay, that you have to write down and then draw the pathway. You have a diagram that you have to draw in your answer sheet. Clear? And include other overviews of hemidegradation pathway. Clear? And there are some significance of hemidegradation and associated disorders also that you need to include in your answer sheet. Okay, that's all regarding short answer. And next is a very short answer. That first one, bile salt. In that you have to mention the name of bile salt and role in digestion. Okay, so mainly I will give you two bile salt that are cholic acid plus glycine, that is equal to glycocholic acid, and second cholic acid plus thiouridine, that is thiourocholic acid. So this two you can mention for bile salt and mention the role of these two. Clear? After that you have to, role means you have to include the functions also. Clear? Then the second question, that is Van de Berg's test. So write down what is the test and what purpose it's doing and the duration and the result, what we can find out from the test. Like that you have to write for the test. Mainly it's used to determine whether excess bilirubin in the blood is conjugated or unconjugated, and therefore whether jaundice in a patient is due to hemolysis or due to any other liver disease. Okay, mainly we are conducting this test for this purpose. Clear? Okay, that is only the two questions, short question and very short question from the hemocatabolism. Okay, we'll move on to the 7th chapter, that is organ function test. From this chapter also we have short answer and very short answer questions. The first one renal function test, second is liver function test. So both these are a diagnostic tests used to detect any disease or any abnormality. So first you have to define what is a renal function test and the indication for renal function test and the components of renal function test, mainly glomerular function and albuminemia. So that's the tests also we are using to detect electrolytes, minerals, protein, waste products, energy sources. Like that other tests also coming in the renal function test. Clear? Okay, and you have to include the clinical significance in that the values sodium, potassium, chloride, bicarbonate and urea, creatinine. All the components, range of values also you have to be mentioned. And normal, next is the liver function test. Liver function test In that you have to, same as in the renal function test, you have to write down what is a liver function test and the indication and the procedure, purposes, and what is the result, management, everything you have to include in the liver function test, and the values. Mainly bilirubin total are there, conjugated bilirubin, unconjugated bilirubin is there, SGPT, SGOT, you may also aware of that, right? So bilirubin total is 0 to 1 and conjugated is 0 to 0.35. Like that you have to mention the values. So for renal function test also there is values. For liver function test also there is values that you have to be studied. Clear? Okay. Then we'll move on to the very short answer question. The first one is mention two enzyme of diagnostic importance in liver disease. So you have to mention at two names. The two names, that one is alanine transaminase and aspirate transaminase. So you can use these two names for the enzyme diagnostic importance in the liver disease. Clear? Then we'll move on to the second question. Normal values of thyroid hormone. Okay, thyroid hormone, we know there are T4, FT4, T3, FT3, TG and TSH. So for this, which purpose we are doing and the values. For thyroid serum we are doing T4 and the value is 4.6 to 12. If free thyroid test, the test is FT4 and the value 0.7 to 1.9, and same like that other tests are there, free thyroid test and serum thyroglobulin, all that the abbreviation is different. Okay, and the value also different, that you have to be studied. Clear? So that are the questions from the organ function test. Short answer question, that is renal function test, liver function test, and very short answer, two enzymes name you have to mention for the diagnostic importance in the liver disease. Second, thyroid hormones normal values. Okay, clear? And next our last chapter, that is the immunochemistry. From this chapter, the expected essay question is type of immunoglobulins. Okay, so for a essay, same as everything, you have to give a definition. What is a immunoglobulin? So write what is a immunoglobulin and the type. What are the type? There are mainly IgG, IgM, IgA, IgD and IgE. Okay, and the structure. You have to name each immunoglobulin and write down separate functions, structures and the properties. Okay, then only you will get the complete score of a question of immunoglobulin. Okay, and in that you can include the functions also. Okay, for IgM the functions, and for IgE what all are the functions, the important, like that also you can include in the immunoglobulin question. Clear? For the short answer question, difference between IgG, IgM, IgA, IgE, IgD. Okay, any two you will get. Maybe you will get differentiate between IgA and IgM, or IgG and IgE. For example, we will take one function. Okay, for IgG, mainly it's a major IgG in extravascular space, and it's a macrophage and monocyte. Also it help to not all subclasses, but it helping for the opponent used to describe the substance that enhance phytocyanosis. Okay, like that you can write the functions and describe in a detailed manner. Okay, and also for IgA, you can write that generally it occurs in mucus secretion including tears, sweat, saliva, colostrum and the secretion of the gastrointestinal, respiratory and genitourinary tract. Like that you can differentiate between to immunoglobulins. Clear? So you have to be by heart all the functions and all the importance of the immunoglobulins. Clear? And next is very short answer. In very short answer, the expected question is 4 advantage of a ELISA test. Okay. For that, you have to define what is ELISA test and the structure, subclasses, function and the serum concentration. Okay, so the definition is IgG refers to a class of immunoglobulin include the most common antibodies circulating in the blood, facilitating phagocytotic distribution of microorganisms, while IgM refers to the class of immunoglobulin of higher molecular weight including primary antibodies released into the bloodstream early in the immune response. Okay, like that you can differentiate between IgG, IgA, and IgD means it refers to class of immunoglobulins expressed in the plasma membrane of a immature B lymphocyte. Okay, so these three you have to define and write down what all are the differences. Okay, like that you can answer for the question. So that's are the question from the immunochemistry. Essay is type of immunoglobulin and short answer is difference between IgG, IgM, IgA, IgE and IgD, and the very short answer 4 advantages of ELISA test. Okay, so these are the expected question. We will just revise all the questions. So we'll move on to our first chapter, that is carbohydrates. Okay, the expected essay is classification of carbohydrates, digestion, absorption and metabolism. Second essay, factors regulating blood glucose level, and short answer, diabetic mellitus, GTT, role of 2 hormones. Okay, and very short answer question is glycosuria, HbA1c and hypoglycemia. And the second unit, the expected essay is digestion and absorption of the lipids, and short answer, functions of essential fatty acid, metabolism of cholesterol, functions of lipoprotein, ketone bodies, MUFA and PUFA. And very short answer question, Rother's test, difference between MUFA and PUFA, trans fatty acid, type of cholesterol, last lipid profile. Moving on to third, the protein chapter. In that essay question, plasma protein, in that classification and biochemical function you have to write. Very short answer, in that digestion and absorption of the protein, amino acid you have to include the classification and importance of amino acid. Okay, third short answer, protein metabolism, and fourth one, electrophoresis. Clear? And from very short answer, essential amino acid, inborn errors of amino acids, proteinuria causes, and last one is a hypergamma-globulinemia. And the fourth one, the clinical enzymology. From clinical enzymology, you will get the essay, probably isoenzyme and the clinical significance. So you have to write what all are the isoenzymes and their clinical significance. Clear? And second is the short answer question. In that you will get the question, that is enzyme in the liver function test. So you have to list down the enzymes and their functions one by one. So very short answer question we can expect, that is the normal values. So you have to by heart the normal values also. Clear? And next, acid base maintenance. From this chapter, the expected essay question is the pH of blood. In that, normal pH, role of lungs and kidney. So the normal pH is 7.35 to 7.45, and how the pH is affecting on lungs and kidney. So that we already discussed. The pH is increased means that is acidosis. If the pH is decreased means that is alkalosis. So going to lungs and kidney means there is several conditions that can cause metabolic acidosis, metabolic alkalosis, respiratory acidosis and respiratory alkalosis, such as COPD, emphysema and chronic kidney diseases, hyperemesis gravidarum, certain conditions that can cause the metabolic and respiratory acidosis or alkalosis. Clear? That you have to be write down in detail. Next is a short answer. From the short answer we can expect 3 questions, that is metabolic acidosis. What is metabolic acidosis? pH is reduced. And second is arterial blood gas and arterial blood gas. Third is the acid base disorder. Clear? And very short answer, the pH definition, blood pH and blood buffer. And from the 6th unit we have only short answer and very short answer question. First one is hemi dehydration pathway, and from very short answer you will get bile salt and the Vandenberg's test. Okay. And the 7th unit, what all are the questions we can expect? For short answer, renal function test and liver function test. Very short answer question, we can expect 2 enzyme and the importance in liver disease. Second, thyroid hormone normal values. Finally, the last unit, that is immunochemistry. From this chapter, the expected essay is type of immunoglobulins, and the short answer, difference between IgG, IgM, IgA, IgE and IgD. Okay, very short answer question, advance of ELISA test. Okay, so that's all regarding the expected questions of applied biochemistry. I hope all are clear. If you have any doubt, you can mention in the comment box. Later videos we will discuss you. Okay, thank you for watching. Subscribe Nursing Guru for further updated videos.